Guys, we are now looking at Staphylococcus aureus toxins. There are 5 toxins you should know. Hemolysin, Panton, Valentine, Leucocidin, PVL is the other toxin. Then you have enterotoxin, toxic shock syndrome toxin, epidermic, what's it, it is epidermolytic or exfoliative toxin. Epidermolytic or exfoliative toxin. So which are the 5 toxins you should know for Staphylococcus aureus? Hemolysin, Panton, Valentine, Leucocidin, PVL, enterotoxin, toxic shock syndrome toxin, epidermolytic exfoliative toxin. First, let us look at hemolysin. Okay, hemolysin you have 4 types. They are alpha, beta, gamma, and delta hemolysins. Okay, these are exotoxins, means they are produced by the bacteria. Endotoxins are in the cell wall, but exotoxins are produced by the bacteria. Okay. Now let us look at alpha lysin. Alpha lysin, also known as alpha toxin, is the most important one. Okay, and it is produced only in aerobic conditions. It needs air, oxygen it needs, and basically it will lyse only rabbit erythrocytes. That is why when you have blood agar, that is sheep blood, right, you will see what? You will see beta hemolysis. Beta hemolysis is very important. Beta lysin, this is hemolytic for sheep cells. The first one, alpha, was for rabbit cells, right? But beta lysin we are going to see in the blood agar. What is that word you are saying? Beta hemolysis. Beta hemolysis you will see in the blood agar, because the blood agar is made up of sheep blood, and beta lysin is the enzyme, or what can you see? It's a toxin. Sorry, it's a toxin that can lyse sheep RBCs. Okay, and this is evident only in cold temperature. It is initiated at 37, but it is evident only at cold temperature. That's like the temperature has to go up till 37 for it to start, but you can visualize it only in cold temperature. So this is known as hot cold phenomena. Please focus on this one. It's slightly important. Hot cold phenomena. It is initiated, the lysis, beta hemolysis is initiated at 37 degrees centigrade, but it is evident only at cold temperature. Okay. So the enzyme is sphingomyelinase C. Fine. Sphingomyelinase C converts the sphingomyelin in the RBC membrane, and hence it is lysed. Okay. So gamma lysin acts on everything. Delta lysin also acts on human, sheep, rabbit, everything. Rabbit RBCs. Okay. Main thing you have to focus on here is the alpha lysin needs what? Aerobic condition. Beta lysin needs hot for initiation and cold to be evident, right? Okay. Let's move on to the next toxin, which is that after hemotoxin it is, after hemolysin it is Panton, Valentine, Leucocidin. So now we are looking at Panton, Valentine, Leucocidin, PVL. It is composed of slow and fast. It will damage the leukocytes and macrophages. So actually I just stopped to check if this is really important, but yeah, they have asked 3 times in RS, so revised scheme, that is RS, if you're wondering what it is. They have asked this question 3 times, so it's better to focus on this. The 5 toxins that you need to know for Staphylococcus aureus are hemolysin, Panton, Valentine, Leucocidin, PVL, enterotoxin, TSST, epidermolytic exfoliative toxin. About this Panton, Valentine, Leucocidin, it has 2 components, slow and fast. The components damage the leukocytes and macrophages. The components, these components, sorry, it has components, 2 components, slow and fast. These damage the leukocytes and macrophages, and it is associated with the MSRA strains. Moving on to enterotoxin, this is responsible for food poisoning. The main MCQ they will ask you is this: within how many hours? Okay, 2 to 6 hours. This is a very important thing they ask you. There are 9 antigenically distinct enterotoxin. A, B, C1, C2, C3, D, E, H and I are antigenically distinct enterotoxins, and type A toxin is responsible in most cases. At least this much you can remember. Type A toxin is responsible in most cases. There are actually how many? 9 enterotoxins. Okay. Then it also resists boiling. If you boil for 30 minutes also, it will not die. This because the bacteria will die, but the toxin will be there. Toxin is produced when Staphylococcus aureus grows in carbohydrate and protein. So basically, even though bacteria dies, the toxin survives. The toxin can act after boiling for 30 minutes. So, I think you should boil for a longer time. What do you say? So, cooked food you should not keep for a long time. Then, the skin of fingers of the food handlers, they should be lesion free. The main source of infection is the food handler's fingers. The toxin is directly going to act on the autonomic nervous system, and enterotoxin is a super antigen. It stimulates lymphocytes in releasing interleukins and tumor necrosis factor. Enterotoxins are implicated in some diarrhea. So some things I want to write here. Enterotoxin is actually a super antigen. Super antigen it is. Enterotoxin is a super antigen. Okay. And does what it does. It's going to stimulate the body to release more of these immune responses, like interleukins, tumor necrosis factor are going to be released more. It is going to stimulate the T lymphocytes to release more interleukins and tumor necrosis factor. Okay. This toxin can be detected by ELISA latex agglutination. Okay. Now moving on to toxic shock syndrome toxin. This is also a super antigen. So enterotoxin also was a super antigen. Toxic shock syndrome toxin is also super antigen. And basically this causes fever, hypotension, vomiting, diarrhea, erythematous rash. Okay, they are most strains which produce this TSST they are belonging to some bacteriophage group 1. Okay. What else you should know here? They also activate T lymphocytes and release interleukins and tumor necrosis factor. This is the same condition, right? It's the same thing which was written in enterotoxin. Do you remember? Please pay attention here. They also activate T lymphocytes to release interleukins and tumor necrosis factor. Now let us look at the last toxin here, exfoliative or epidermolytic toxin. Here what happens is there are 2 types of toxins, A and B. Okay, and A is heat stable, it can resist heat, and the production of the toxin A is under the chromosomal control. However, toxin B is plasmid, it's regulated by the plasmid. Okay. So what are these going to cause? They are going to cause the staphylococcal scalded skin syndrome. This is very important. SSSS, staphylococcal scalded skin syndrome. Okay, it is also known as Ritter's disease in newborn. Okay, milder forms are called pemphigus, neonatorum and bullous impetigo. All these are related to staphylococcus. Please pay attention here. This is the diagram. The Ritter's disease will be a severe form. Scalded staphylococcal syndrome, right? Skin. Staphylococcal scalded skin syndrome. And this is pemphigus neonatorum and bullous impetigo. These 2 are milder forms. Okay. And all these are caused by what? Exfoliative or epidermolytic toxin. Fine. So this was a long video. You have to learn about hemolysin, alpha, beta, gamma, delta, Panton, Valentine, leukocidin, PVL. This is having two types, slow and fast, and it is going to damage the WBCs and macrophages. Enterotoxin is also a super antigen. TSST, that is toxic shock syndrome toxin, is also a super antigen. Epidermolytic exfoliative toxin will cause you the staphylococcal scalded skin syndrome, right? See, in this hemolysin, beta hemolysin, you saw hot cold phenomena, okay? Then in PVL, you saw slow and fast components. Enterotoxin, there is type A enterotoxin, which is causing more of these food infections, and 2 to 6 hours is within 2 to 6 hours the person will have reaction. TSST is also super antigen. It is one second. So even enterotoxin is super antigen, it's added there, and this is TSST toxin is produced by bacteriophage type. So it is actually bacteriophage group 1. Okay, that strain will have this toxin. Okay, epidermolytic or exfoliative toxin will cause staphylococcal scalded skin syndrome, or the pemphigus neonatorum, bullous impetigo, milder forms. Then you have the Ritter's disease, which is more severe, Ritter's disease. Please note one thing here, this toxic shock syndrome toxin 1 colonized the vagina. That's why people who used to use tampons earlier, that time it was unknown, they used to have this toxic shock syndrome. Okay.